Everyone for joining online and for the people that are here in person. Thank you. And today we're going to have a great talk from Sarah, but I will begin with a land acknowledgement. We acknowledge that we live and work on the Coast Salish peoples of this land, the land that touches the shared waters of all tribes and bands within the Suquamish, Tulalip, and Muckleshoot nations. And we also acknowledge that we work and live on the Duwamish people's land, the first people of Seattle. So today we're going to hear from Dr. Sarah Saunders, and she received her PhD at the University of Minnesota, working with Dr. Francie Cuthbert, where she studied the environmental and genetic factors influencing survival and reproduction of Great Lakes piping plovers and endangered shorebird. And most recently, she conducted postdoctoral research with Dr. Elise Sipkin at Michigan State University. And while there, Sarah modeled the abundance, distribution, and climatic drivers of monarch butterfly populations using citizen science data. She joined the National Audubon Society in 2018, where she is currently a quantitative ecologist in the science division. And Sarah primarily works on research projects focused on the Great Lakes region, including modeling occupancy and abundance trends of marsh birds and water birds, and coordinating science work across Audubon's Great Lakes Initiative. So thank you, Sarah, and you can take it away now.
Great. Thanks, Bria. Thanks for inviting me, and thank you for everybody for tuning in if you're online and for being there in person, and happy Earth Day. It is Earth Day. So I'm a quantitative ecologist, as Brielle said, at National Audubon Society, and I'm in the science division, but our whole team works remotely, and so I'm based in my home office in Grand Rapids, Michigan. And I'm excited to be here today to talk about integrated modeling and integrated population models in particular. And while I consider myself an ornithologist and all the work I'm going to talk about today is focused on birds, don't worry, I think you'll see a lot of applicability outside the world of birds. And actually, IPMs were first introduced in the world of fisheries. So I originally titled this talk, Disentangling Data Discrepancies and Evaluating Population Viability Using Integrated Population Models, which is quite a mouthful, but I wanted to make sure I captured the breadth of what IPMs can do. And as I was putting my talk together, I realized that I can summarize the power of IPMs in a more succinct way that is still just as impactful. So IPM's a powerful tool for biodiversity conservation. And in the course of this talk, I hope I will be able to prove to all of you that this tagline is actually true. So I'm going to start with defining what we mean by data integration, and then really break down the steps for building an integrated population model. And then I'll go into the power of IPMs for conservation, which I'll highlight with a few case studies on birds, so bear with me. And then I'll conclude with some recent advancements of IPMs and remaining opportunities. So what is data integration and why do we do it? We all know that biodiversity loss is one of the most pressing environmental problems, and it impacts ecosystem functions, community dynamics, and human and wildlife health. So understanding the impacts of anthropogenic change on wildlife populations requires research at large spatial and temporal scales. But in an era where macro scale conservation needs are not met due to limited research funding, it's often not possible to be able to obtain necessary population level and demographic data from a single collection effort. Even for threatened species that have restricted ranges, collecting data and estimating those ecological reasons for declines is challenging because such species tend to be rare or elusive, which makes obtaining sufficient data for those standard population models difficult. In addition, species are often widely distributed across heterogeneous landscapes. And this is especially true for migratory species, such as the monarch butterfly, which is shown here on the left. And the monarch butterfly uses the whole of North America throughout its annual cycle. But it's also true for many other species. There might be a number of different factors influencing the dynamics and underlying demographic processes of a population across the full annual cycle. which can influence how local abundance varies across a species range, so that different proportions of the landscape contribute unevenly to population persistence. And then finally, climate and land use changes are occurring at unprecedented rates. So what may constitute good conditions now won't necessarily be so in the future. So data for particular populations or entire species are frequently incomplete or available from disparate data sources that are collected under differing protocols, or of variable quality due to different levels of efforts over time and space. You can see that again here on the left for monarch butterflies. You can see there's a lot of data availability during the summer season, which is shown there in yellow, and comparatively less in terms of data availability during the winter and spring seasons, shown in blue and green. So these conditions result in limited longitudinal data to model population viability, extrapolate inference at large scales, and detect significant changes in population trends within timeframes for appropriate management actions. So as a result, understanding the population dynamics of threatened species, and indeed most species, is typically either inferred through independent local scale studies or achieved through large-scale niche modeling that is correlative, with limited power to elucidate the underlying mechanisms. But relatively recent integrated modeling approaches that combine multiple data sources across spatiotemporal scales into a single analysis provide a foundation for overcoming these problems, and they allow research to scale from the local biological processes to regional level patterns where management occurs. So data integration can refer to many different types of methods, including even just integrating many different data types into a single database. But here we're going to talk strictly about data integration in the form of modeling, also known as integrated modeling. And in particular, I will dive into case studies specifically using integrated population models. But generally, integrated modeling refers to the incorporation of multiple data types on a single target organism into a single modeling framework. So typically the data that we use in integrated models were not actually collected with the idea that all of them would be analyzed together. So you may all know that the concept of integrated analyses within ecology was first formulated in fisheries science in the early 1980s. And since then, there have been variations on these approaches within a variety of subfields. So what exactly are integrated population models? Well, integrated population models, or IPMs, are a well-developed subset of integrated models in which the analysis of a joint likelihood of two or more data sets allows for the simultaneous estimation of both population abundance and the processes leading to population change. So IPMs provide an approach for addressing issues of sparse or fragmentary population data by incorporating multiple data types like census, productivity, and telemetry data into a single dynamic model of the target population. And IPMs typically include the analysis of data on population abundance, trajectory, or structure. So that comes from data types like census or count data, and then data on demographic parameters like productivity and capture mark recapture data. So a key feature of IPMs is the ability to model a population's underlying dynamics, including important life history features, and by explicitly accounting for the processes that lead to population changes over time. So in other words, birth, death, immigration, and emigration. So the objective of IPMs is to incorporate as much mechanism as possible to estimate a population's trajectory and assess the primary factors causing changes in abundance and demographic rates. So by incorporating all available data on a population, IPMs can overcome limitations of traditional independent analyses and allow for the simultaneous estimation of demographic parameters and population trajectories, while also standardizing the error structure across those different data types and accounting for multiple sources of uncertainty. They also allow for the direct linking of variation in population abundance to changes in stage-specific vital rates and covariate effects. So the hierarchical structure of IPM separates the underlying and unobservable ecological mechanisms from the observation process. So that allows for disentanglement of process variation from sampling variation. And this is typically done in IPM through a state space formulation, which assumes that abundance may be over or undercounted. So here I'm showing you a state space formulation of the Cormack-Jolly-Sieber model, which is used to estimate survival by following marked individuals over time. And you can see that the true state of the individual, alive or dead, is shown in that top row, and the observed capture history is shown in the bottom row. And this separation of the state and observation processes allows us to account for potential biases due to imperfect detection during sampling. So failure to accurately account for the sampling process can lead to misleading or erroneous inferences on population status. And it's especially important to consider sampling methods in situations in which detection varies spatially or through time, which you can imagine occurs pretty frequently. So IPMs are built according to 3 general steps. In step 1, a population model linking abundance and demographic rates is developed to capture the important life history features of the target species. And this is typically a stage or an age structured matrix projection model, where the population size of each stage or age class is assumed to be a function of the population size in the previous year and the demographic rates. So here I'm showing an example from the common loon case study that I'll go over in a little bit more detail later in the talk. But for this, for our latent population model for loons, we assumed that the minimum age at first breeding was 4 years old. And annual apparent survival was constant for birds 3 years of age and older. And stage-specific abundances were allowed to vary according to stochastic processes. So for example, the number of fledglings here and not is a function of the number of breeding adult females in a given year and their fecundity. The number of new 3-year-olds follows a binomial distribution defined by the number of fledglings from 3 years prior and the juvenile survival rate, which represents a mixture of survival from fledgling to age 3 and transition to the adult stage. So this parameter encompasses several years because 1- and 2-year-old loons do not return to the breeding grounds, so they're essentially unobservable. That's why they're shown here in gray on the life cycle diagram. And then finally, the number of breeding adult females each year follows a binomial distribution defined by the number of 3-year-old females from the previous year, last year's breeding females, and the adult survival rate. And we can also incorporate environmental stochasticity by allowing vital rates to vary annually using random temporal effects, as well as include covariates with appropriate link functions to incorporate any relevant biotic or abiotic factors that may influence demographic rates or detection probability as well. So in step 2, the likelihoods of each data set are formulated to estimate the parameters defined in the population model. So in this diagram, the parameters to be estimated are shown as orange boxes: fecundity S, population abundance N, and then juvenile and adult survival rates shown with B. The likelihood functions, which are specified independently for each data type, describe the probability of an observed outcome, which are the data shown in blue triangles, which are conditional on particular parameter values. So for example, capture-recapture data, or M shown here, can be analyzed using Cormack-Jolly-Sieber models, which generally use a multinomial likelihood to estimate survival and recapture probabilities. So that recapture probability is shown here in green, PM, as is the observation error associated with the count data, Y. So a state space likelihood, one in which the true demographic process is modeled separately from the observation process, is often used for time series data like the count data. And a Poisson regression model is typically used for reproduction data when they consist of counts of younger fledglings, which is shown here as NF, constrained by the number of breeding adults, NA. And it's essential that the component likelihoods for these different data types have at least some parameters in common, as you can see here by the overlapping boxes. because that's the mechanism by which information is shared across the data sets in the IPM. So finally, the joint likelihood of all data sets combined is specified, typically by multiplying the individual component likelihoods that were defined in that second step. So in addition to the assumptions of each of the component likelihoods, an implicit assumption of IPMs is independence among likelihoods and data sets. So in the strictest sense, this implies that animals seen in one data type cannot also be observed in the other data types, but this rarely occurs in practice. And while lack of independence among data sets can lead to overconfidence in parameter estimates in the form of inflated precision, several recent studies have actually demonstrated that partial or complete dependency among data sets has minimal consequences on IPM estimates. So here I'm showing results from one of those studies, Wiegman et al., that just came out last year, where estimates from a completely independent model, which are shown here in blue, are shown against a partially dependent model in pink. And you can see that the like-for-like estimates for each parameter are remarkably similar, except for maybe with the exception of immigration rate, which is shown in the bottom right panel. Those ones appear to be a little bit more different, and that's typically not unexpected for immigration rate, which is the hardest parameter to estimate given that there's not explicit data. But it's important to note that this result may not hold true for every data type combination and IPM structure. So once the joint likelihood is specified, the IPM is analyzed to estimate the parameter values. And the hierarchical structure of IPMs naturally lends them to Bayesian analysis, although the joint likelihood can also be analyzed using frequentist inference as well. But Bayesian analysis has become a common method for describing uncertainty in both fisheries and wildlife management. So I'm guessing you all are familiar, and it facilitates the inclusion of additional information in the form of prior distributions. And prior distributions can be uninformative or informative based on information from previous studies on different populations or similar species, as well as meta-analyses or even expert opinion. And building an IPM and a Bayesian framework allows for the most efficient use of available data and the best possible description of uncertainty. And it's also the most rigorous method for incorporating parameter and demographic uncertainty into forward projections. So to successfully manage populations for conservation purposes, it's necessary to track changes in demographic parameters, identify vital rates having the greatest influence on population growth, as well as determine the factors driving spatial and temporal variation in those key rates, and whether or not those relationships are actually stationary. And IPMs are especially valuable in the study of declining or small populations because gains in accuracy and parameter precision are most apparent in complex models with large numbers of parameters. And that's often the case for conservation-focused projects encompassing multiple interacting threatening processes. So as such, IPMs are well suited for wildlife conservation research, providing a powerful tool for holistic analyses on threatened populations, as I'll demonstrate with a few case studies. So by using all available data that contain both direct and indirect information about demographic rates, the IPM framework allows for estimation of parameters that would otherwise be difficult or impossible to evaluate if data were analyzed separately. So in particular, IPMs can provide information on the sources of population level gains, like immigration versus recruitment, without explicit movement data. And quantifying the relative contributions of immigration versus reproduction to population growth allows us to identify the relevant spatial scale for effective conservation and appropriate action. So for example, if local level reproduction has strong effects on population dynamics, the spatial scale of dynamics and the required conservation actions will also be local. But if population dynamics are more strongly influenced by immigration, the scale of management depends on the extent to which local populations interact and the rate at which individuals migrate. And that typically necessitates conservation planning in a much broader spatial scale than that of the target population. So an example I'm showing here Reichert et al. Demonstrated a disproportionately large role of immigration in facilitating population growth of a snail kite population that was centrally located in the species range. So it was essentially acting as a connectivity hub. And the results provided much needed information on the role of connectivity in population growth, suggesting that conservation efforts focused explicitly on centrally located habitat patches and small populations are likely to have the greatest benefit for the species. So as another example of this from some of my work, I estimated immigration with an IPM for Great Lakes piping plovers, an endangered shorebird population, which is circled here on the map. And the inset shows some of the recent nesting locations throughout the Great Lakes region. And this population is heavily managed given its endangered status. And we were interested in understanding the extent of immigration into the population from the two other plover populations that are shown here in blue, in the Great Plains to the west, and then the Atlantic coast to the east. And both of these populations are larger, so they're listed as threatened. So if there is exchange of individuals between these populations, it would have important implications for how each of these populations are managed, especially if there was a high rate of exchange between them. So what did we find? We estimated that immigration during the 25-year study period was low, with an expected number of only about 2.6 females entering the population annually on average, which is shown there as the solid pink line. But we did find that immigration varied considerably across years, with as many as 7.8 females estimated to have entered the population in 2002. And you'll notice that the precision on these annual estimates is rather low, which is unsurprising given that we did not have explicit movement data by which to estimate these rates. But nevertheless, estimation of immigration does become possible when count data are combined with productivity and recapture data as in the IPM. Because count data intrinsically contain information about all demographic processes in a population, allowing for extraction of additional information on immigration rates when we model it with other data sources. And these findings of relatively low immigration rates and its correlation with population growth rate being really low, which is shown here on the right, indicated that piping plover conservation efforts focused on achieving high productivity and pre-fledging survival at the local site level should be prioritized over broader management actions to promote exchange of adults between populations, such as through restoring unused sites to attract new breeders. So another capability of IPMs is the ability to estimate population viability within a Bayesian framework, which is called Bayesian Population Viability Analysis. And by combining parameter estimates informed by multiple data sets into a single likelihood, the uncertainty around all demographic parameters is accounted for and propagated into projections within a unified modeling framework. So to make predictions, we simply augment data sets with NAs for the number of future years for the population survey, and then the demographic rates and population abundances are projected as part of the model fitting process. We can then assess population viability by calculating the cumulative probability of extinction or quasi-extinction, as well as other derived quantities based on those posterior distributions, like the probability that the population size in the last year of the projection window would be greater than the size estimated during the last year of the observed data. So here I'm demonstrating this again for piping plovers, where you can see that the IPM BPVA framework allows us to estimate demographic rates retrospectively, so to the left of those dashed lines, as well as prospectively, shown to the right of those dashed lines. The uncertainty around the estimates is shown as a 95% credible interval in gray. And as you can see, the uncertainty gets much more pronounced as we project farther into the future. So shorter projection windows, like the 10 years that I'm showing here, are more meaningful because uncertainty just tends to get too great beyond this time window. So using this projection framework, it's easy to see how we can adapt it to evaluate options by comparing both predicted cumulative quasi-extinction probability and total population size in 10 years. So again, we did this for plovers because you're interested in evaluating management of various sources of predation pressure on the population. So for the null scenario, we forecasted population sizes and vital rates using the method I just described. And then we compared the null scenario to the following hypothetical management alternatives. Increased control of chick predators, that includes crows, gulls, and mammals, with the effect of a 20% increase in the number of fledglings, or FT. The second scenario was increased control of merlins, which is their main avian predator. We have reduction in their annual abundance by about 20%, which was included as a covariate XT on adult survival. So this is essentially an effect of increasing annual adult plover survival rates. And then we looked at simultaneous effects of both chick predator and merlin control. And we specifically designed our management scenarios to reflect realistic conservation considerations that could be implemented by Fish and Wildlife Service with the goal of helping them to inform their strategies going forward. So as you can see, estimates of quasi-extinction probability, which we defined as 15 breeding pairs remaining at the end of that 10-year projection window, differed among these four scenarios. So the chick predator control scenario reduced quasi-extinction probability from 11.9% under the null scenario to 8.7%. And the merlin control scenario more than halved the probability to 3.5%. And then combining both of these control strategies resulted in the lowest quasi-extinction probability of 2.6%. We also calculated that simultaneous implementation of both predator control scenarios was the only option that produced a greater than 95% probability that the resulting population size would be larger than under no proposed management. So these findings told us that piping plover conservation efforts should include development of effective selective control strategies that target the predators most strongly impacting productivity and survival rates on a site-by-site basis, rather than following the ad hoc protocols that were previously in place. So for example, certain nesting sites have consistently low adult survival due to heavy merlin predation pressure, whereas others have consistently low chick survival due to high concentrations of chick predators. So focusing on improving survival rates during both of these life stages is the approach most likely to maximize population recovery and subsequent delisting. So in addition to evaluating management scenarios, I've used the IPM BPBA framework to evaluate potential climate change impacts on common loons breeding in Wisconsin. So in this study, we assessed climate and land use impacts on the demographic rates and population dynamics at their southern range margin over a 25-year period. And that's shown with a star on the map below, overlaid with their breeding range shown in pink. We also projected survival and reproductive success 10 years into the future under a suite of climate and land use change scenarios. And we estimated population viability in response to projected global change, while simultaneously accounting for environmental and demographic uncertainty. So we tested for the effects of quite a few local and broad-scale climate and land use covariates on adult survival, juvenile survival, and fecundity of common loons. And we not only wanted to evaluate environmental conditions during the breeding season when the data were collected, but also outside of the breeding season, given that we're estimating annual population dynamics within the IPM. So we essentially wanted to have a full annual cycle perspective when assessing impacts on this loon population. And I won't talk about each of the variables that we tested, but we did follow a variable selection procedure, and the resulting top supported model included effects of summer rainfall, the winter North Atlantic Oscillation, or NAO, and surrounding developed land cover, all of which are shown here in yellow. And for those, you guys are probably very familiar with the NAO, but for those who don't know, the NAO is a driver of climate variability that has been shown to affect the marine environment and ecosystems along the eastern coast of the US and Canada, where common loons from the upper Midwest overwinter. And because large-scale climate oscillations often produce lagged effects, especially for long-lived species, we considered NAO covariates at time lags of 0 to 3 years. So we used these results of significant covariates from our retrospective analysis to project loon population viability into the future. So we assessed common loon population viability by projecting the number of breeding pairs 10 years into the future as part of the model fitting process, and evaluating the population under future global change scenarios. So for all scenarios, we used annual projections of summer rainfall in developed land cover that were available, but projecting NAO indices is notoriously difficult and is typically only done on seasonal or decadal timescales. So we designed 6 scenarios that varied the winter NAO annually based on the long-term and short-term means and trends of the index. We incorporated that as informed prior distributions for future years in our analysis. So that goes back to the slide I showed earlier, where we're opting to provide an informed prior distribution to estimate how the population will respond under particular scenarios in the future. So what did we find based on these scenarios? Well, in general, the mean and long-term NAO trend scenarios, which I'm calling A through D in that top row, yielded similar projections of adult survival and fecundity, as you can see to the right of these dashed lines in each figure in the middle and bottom row. So again, these are NAO scenarios based on the long-term and short-term means, which are shown in A and D, and then B and C are the scenarios based on a weak positive or a weak negative trend of the index. You can see that with those thick brown lines in each panel in the top row, and those thin lines indicate 10 random MCMC iterations to illustrate the variation around those averages within the Bayesian framework. So under all of these scenarios, we're seeing similar population projections because these vital rate projections are similar under each of these NAO scenarios. However, that wasn't the case with every scenario that we looked at. So you're seeing scenario E here, the short-term positive NAO trend scenario, and that resulted in the lowest mean estimate of adult survival, but the highest mean estimate of fecundity in 2029, which was that last year of our projection window. On the other hand, the short-term negative NAO trend scenario projected a stabilization of adult survival over the 10-year period, but it resulted in the lowest mean estimate of fecundity. And overall, the lowest loon population sizes were predicted under positive NAO trend simulations like scenario E. And that is a pattern which is anticipated to occur with ongoing climate change, raising concerns about the possible extirpation of this population along their southern breeding margin. So our work demonstrates that interactions between climate variables results in complex patterns of population dynamics that may take multiple years to become apparent in a long-lived high trophic level species, as evidenced by the support of a lagged effect of NAO on both adult survival and fecundity of common loons. And importantly, our findings tell us that considering environmental conditions throughout the full annual cycle is critical for accurately assessing viability, especially for those populations along species trailing range edges. Okay, the next case study I want to talk about focuses on disentangling data discrepancies with IPM. We all know that a common challenge for studying wildlife populations occurs when different survey methods provide inconsistent or incomplete inference on the trend, dynamics, or viability of a population. And inconsistencies could be introduced within the data collection process, like non-random sampling in space or among ages and stages within a population. Other discrepancies are the result of spatial or temporal variability in demographic processes across geographic and/or temporal extent. So for example, census data from the North American Breeding Bird Survey, the Audubon Christmas Bird Count, and Migration Watch sites suggest a recent decline in abundance of American kestrels, which are shown there on the left. But population growth rates derived from these censuses vary across the subspecies range. In other systems, cross-scale interactions of both spatial and temporal variability may contribute to inconsistent population trends. And that's exemplified by the Eastern North American monarch butterfly population, where census data on monarchs at their overwintering grounds in Mexico indicate a 19-year downward trend, but trend estimates from count data at their summer Midwestern US breeding sites have been inconsistent. And issues of spatial scale mismatches may also lead to dataset discrepancies, given that fine scale indices may show contrasting trends compared to large scale indices. So for example, bobolinks on the right there, a grassland bird of conservation concern in North America, experienced significant provincial level declines across much of their breeding range in Canada. However, recent work demonstrated that a majority of local bobolink populations have not strongly declined at finer scales over that same temporal interval, suggesting a more optimistic trend than that obtained from analyses at broader spatial scales. Yet we can exploit the capabilities of IPMs to address inferential discrepancies that stem from spatiotemporal data mismatches. So we illustrate this issue with analysis of a migratory species, the American woodcock, in which individual monitoring programs suggest differing population trends. So continental scale surveys of displaying male American woodcocks indicate declines of approximately 0.6 to 1.1% per year in the central and eastern populations. However, estimates of annual population change based on fecundity and survival rates that are measured from harvest and banding data suggest that woodcock population should be declining by about 17% per year. So without addressing this discrepancy, we cannot fully understand their range-wide dynamics, nor we can develop robust management strategies, which are especially critical for a harvested species. And although there have been analyses of American woodcock population dynamics based on individual data sets, prior to this analysis, there have been few attempts to combine these data and no attempts to use an IPM for a population level analysis. So you can see with the life cycle diagram on the right that integrating these data sources that are collected at different times of their annual cycle and in different regions was rather complex, but necessary given that we really wanted to leverage the information sharing capability of IPMs, as well as their ability to reduce biases inherent to each data source without omitting any of the seemingly contradictory data sets. So I'm going to skip the details on the modeling in the interest of time, but please check out the paper for more details if you're interested. So what do we learn about disentangling these data discrepancies? Well, combining all woodcock data into an IPM allowed us to resolve apparent discrepancies and determine that both the central and eastern populations have declined at an average rate of approximately 1% annually since 1963, which is consistent with the long-term monitoring data from the surveys of displaying males. We were also able to attribute these declines to juvenile and female survival in both populations, and fecundity, particularly in the central population. And our IPM also revealed that the eastern population has stabilized in the last few decades, as you can see there on the right, while the central population has continued to decline. And those are results that have not been documented with analyses of any of the individual data sets. They demonstrate the importance of accounting for spatial variability. And temporal and spatial variability in woodcock monitoring methods and effort likely also contributed to discrepancies in trend estimates. So for example, half of all banding effort in the eastern population occurred during a 7-year window, and 70% of all banding data for the central population came from Michigan, as you can see in this map on the right. So long-term survival estimates based on analysis of band recovery data alone may be informed by data that are not spatially or temporally representative of the entire population and time series. So data availability issues may be the underlying cause of the discrepancies in population inferences that we get from analyses of independent data sets for the species. So in many instances, like our woodcock case study, simply combining all available data sources into an integrated modeling framework will be enough to resolve discrepancies that may arise from analyses of individual data sets. But IPMs can also be used within an adaptive monitoring framework, such that future data collection efforts can be guided in a way that reduces sources of uncertainty. So vital rates that are estimated with low precision, for example, adult female non-breeding survival in the case of woodcock, can be the focus of spatially and temporally targeted studies that can test the validity of IPM-based predictions and allow for model updates going forward. So the last case study I want to touch on relates to estimation of population synchrony, or coincident changes in the size of local populations, which is a common ecological phenomenon exhibited by many taxa. Synchronous dynamics over a species range can be critical for conservation as it increases the extinction risk of spatially structured populations. But little is known about the relative effects of demographic rates on synchrony because of difficulties obtaining data, estimating rates independently from all populations, and making spatial comparisons. But you guessed it, an IPM framework is particularly advantageous for analyzing data from geographically distinct populations because it's possible to handle differing sampling protocols and survey periods among sites, enabling conservation management to scale from a single population to an entire metapopulation or species. So here I'm showing this for Blackturns, which is ongoing work in collaboration with Kayla Davis, a PhD student at Michigan State. And for the study, we were interested in comparing demographic rates among 3 Blackturn colonies, 2 in Michigan, St. Clair Flats and Wigwam, and 1 in Ontario, which is called Tiny Marsh. We had different time series of data for each site, as you can see, and different data collection protocols that we had to account for. So we're able to compare fecundity at the 2 Michigan sites and adult survival across all 3 sites. And you can see here that although the annual estimates differed somewhat across sites, there are remarkably similar patterns over time, with fecundity increasing in recent years at the 2 Michigan sites, likely due to changes in Great Lakes water levels, and then a similar dip in adult survival in 2020 at St. Clair Flats and Tiny Marsh. And we're currently working on adding an environmental covariate to try and reveal possible shared mechanisms behind these annual fluctuations. Because determining the degree and causes of synchrony within metapopulations provides an opportunity to adjust management strategies accordingly. And that will be crucial for Blackturns since they're undergoing severe population decline. So I also want to note that recent IPM developments have expanded these approaches to a multi-species context, facilitating enhanced understanding of spatiotemporal changes in community dynamics and population synchrony among species. So a 2017 study demonstrated this by integrating data along 2 different dimensions, across species and within species using IPM. And the authors analyzed data from 3 seabird species breeding on the Isle of May by combining demographic data sets with island-wide population counts to construct the first ever multi-species integrated population model. And you can imagine that this framework could be extended even further by adding another hierarchical level of colony sites so that you can examine synchrony both across species and colonies, essentially creating a multi-species meta-community IPM. The possibilities are pretty endless with IPM. So speaking of possibilities, that brings me to some other recent advancements in integrated population modeling and remaining opportunities. So development of IPMs using spatially explicit capture-recapture data extends the utility of conventional IPMs by accounting for spatial variation in demographic, movement, and detection parameters, and enabling the ability to make spatially explicit predictions of abundance or vital rates. So Chandler and Clark developed this advancement where the basis of the spatially explicit IPM is a spatial point process describing the number and locations of individuals in a population during the initial time period, and the variation in abundance over time as a function of depth, recruitment, and movement. And both capture, recapture, and survey data can be modeled conditional on the point process. And the model provides a straightforward means of predicting abundance or density in unsampled regions and time periods, and can be used to map and project species distributions and vital rates at broad spatiotemporal scales, which is particularly beneficial in conservation-related projects where logistical constraints frequently prevent survey coverage across a species' entire range or over long time periods. So here I'm showing a result from Chandler et al. 2018, where they used a spatially explicit IPM to get spatially explicit population growth rates for Canada warblers in North Carolina. And you can see they can also illustrate the spatially explicit uncertainty around those estimates with the upper and lower credible intervals shown in those top and bottom panels. So another recent advancement of IPMs relates to incorporation of both sexes into the modeling framework. The typical IPMs are female-based models, so only female life history is specified for the state process. They frequently do not account for mate availability, differing sex ratios, or polygamy among individuals. But two-sex models are required when vital rates significantly differ between males and females. Males are the limiting sex in the population, or when life history characteristics vary by sex, such that modeling the dynamics of a single sex is inadequate. And this is often the case for at-risk carnivore species with polygamous mating systems. And recent IPM advancements, like that developed by Tanan et al. In 2016 for brown bears, allow for the investigation of population dynamics and quantification of cause-specific mortality rates in a non-monogamous species, enabling the use of IPMs in a whole new suite of mating systems. So in this framework, the state process is specified using a two-sex population model in which the annual number of breeding individuals is determined by explicitly modeling the probability that males and females breed as a function of the availability of opposite sex individuals. So what remains for investigation in the world of IPMs? Well, there's always more and different data types to include. Citizen science data are well suited to integrated modeling because their wide geographic distributions of volunteers can provide much richer data sets than are typically available. And the prevalence of these programs has surged in the last few years. So the addition of eBird or breeding bird survey data to an IPM, for example, has the potential to reveal large-scale population trends, and particularly shifts in population abundances and demographic rates at leading and trailing range edges. In addition, data from camera trap arrays can be incorporated into IPMs like Mark V sighting data, which expand the scope of inference for particularly elusive and secretive species like large carnivores. An incorporation of other unmarked data types into demographic estimates is also needed, which can be done through inclusion of recently developed submodels for time series or census data that are modeled according to a Markovian process, whereby individuals remain in the population through a stochastic survival process and are gained to the population either via recruitment or immigration. So I know I covered a lot here, so here are a few of the take-homes I would like to leave you with. Record species losses have hastened efforts to identify extinction risks and ameliorate the ultimate causes of decline. And although characterizing population dynamics and trends is challenging, the spatial and temporal scope of ecological research and conservation efforts has expanded greatly over the last few decades. This then requires ever more efficient data collection and analytical methods, such as integrated modeling, of which integrated population models represent a powerful approach. The development of IPMs overcomes many drawbacks associated with classical modes of inference and has advanced our ability to investigate environmental and demographic drivers of population change, as well as more accurately estimate population trends. The key strength of IPMs is that each submodel borrows information from other submodels, making use of all available data from a study system, leading to the best possible inference on a target population. There are a lot of resources on IPMs out there, and I want to be sure to mention the newly released IPM textbook by Mikkel Schaub and Mark Carey. That's the most comprehensive guide to date on IPMs. And importantly, it includes many more examples and extensions that I didn't cover here, but that can be applied to virtually any study system or a population to help improve recovery for threatened or declining species worldwide. So I encourage you to check it out. So with that, thank you for listening, and thank you to the seminar committee for inviting me, and I'd be happy to take questions. And I've included my email here too for anything to follow up about.
Great, thank you so much. Hopefully you can hear people clapping in person. Yeah, so thank you for that. Yeah, it was a good kind of primer to integrated population models, and your 2019 paper has helped me, so I hope other people will read that online, and people in person will check that out as well. Yeah, so as a reminder, people in person and online can post questions. Yeah, we can just wait a minute when questions come in, but I have a question to start with. So I was wondering, with your plover study, why did you have 20% for like the predation values for both of them? Like, could they, do you think there might be differences between them, or even when you add them together, you had 20 and 20 for both of them? Do you think maybe something with incorporating both of those might have different values? Yeah, I was just wondering about that.
Yeah, that's a good question. We really worked with Fish and Wildlife Service to kind of get a sense of, is 20% feasible for potentially reducing the marlin population annually, for example. So it was really helpful to actually have that connection with those managers to help decide on those numbers. And in addition, we also kind of did a sensitivity analysis, so we didn't actually incorporate into the paper where we messed with that value more than just the 20% to kind of get a sense of like how much is this impacting the predicted population size in the future, as well as population viability. So if you don't have that like connection with managers, it would be really interesting to kind of expand the suite of potential scenarios that you investigated by trying multiple different changes to those vital rates.
Cool, thank you. All right, we have a question from Elizabeth Holmes. Elizabeth says, on the slide with the central and eastern trends for males and females, it looks like the sex ratio was changing a lot for the eastern population. Know what would drive that?
That is a good question. Yeah, one thing, we did incorporate harvest data into the IPM, but we didn't really dig much into the sex ratios of harvested individuals between the central and eastern populations. And so some changes in terms of like harvest and management over time between the two populations may be contributing to those differences in sex ratios. And then we also had something in the supplemental information that kind of dug into that a little bit more in terms of like, are we seeing more males and females banded necessarily in these populations too? So as someone who is outside of like the Woodcock biology world, I don't have an actual biological explanation for you, but yeah, that's definitely something that kind of became a flag from our analysis when we passed it over to the managers. Our results, they picked that up as well.
Cool, thanks. All right, so there's no other questions. Oh wait, just one popped up. Cool. So Alan Hicks says, thank you very much. Very interesting and informative talk. There are a lot of parallels with fish stock assessment. Have you considered methods to weight the individual likelihoods when combining into a total likelihood? One method is to simply use the variance from the data, but fish stock assessments have been investigating other weighting methods.
Yeah, that's something that I actually haven't delved too much into, but we do discuss in actually the Woodcock paper. I think that came up as a possible future direction of how do you weight likelihoods based on, for example, your confidence in the data quality that goes into the integrated population model. So we talk a lot about that in the bird world too. So if you're integrating like eBird data with structured survey data that's collected formally by scientists, like do you want to weight that structured data more heavily than the eBird data just because that's less information rich and potentially more biased? So yeah, that's something I would like to learn a lot more about, like how do you make those decisions in terms of weighting likelihoods based on quality of data, for example. So
yeah, cool. Ellen says thanks. I guess I have a follow-up question to that. I was wondering if with like apps like eBird and other stuff like that, I don't use it. I probably should, but I'm wondering if maybe it can be improved by doing something like the user identifies whether they're kind of a good at birding versus bad at birding. Have you like noticed that in any of those types of data sources? Or yeah, I was just wondering if you can talk more about the citizen science data challenges.
Yeah, that's definitely one of the challenges, is trying to understand, you know, how citizen science is biased by the skill of the observer. And, you know, Cornell that, you know, run eBird, they have some standard methods for accounting for that. There's so many eBird observers that you can't like necessarily give it an individual identifier and account for that in that way, just because there's millions and millions of users. But there's standard like filtering protocols that you do before you can incorporate data into analyses. And that reminded me of something else. Oh, and then also on eBird, you can indicate whether the observation you're submitting is part of a complete checklist, which is really helpful, because then you're getting a sense of, you know, if you're recording all the birds that you're observing, then you can fill in zeros essentially for those species where you know they went to the location and they did not see that species, as opposed to just assuming they didn't report it when they were in fact looking for that bird species. So that's been really helpful too with incorporating citizen science data, because you can kind of backfill the zeros that way.
Yeah, that's super interesting. Yeah, I wonder if there's any issues with using eBird with like days like the bird days or like the holiday count days, where you get like inflated counts because everyone's out versus like an average Saturday. I bet that's probably a challenge too. But
yeah, they have some standard like spatial and temporal filtering too. That's like, if you throw down like a standard grid on a map, for example, to only take one random complete checklist from that grid cell, because then you do have like pseudo replication essentially if you for some reason are looking at a grid cell that has a lot of interest to eBirders. So yeah, that's another issue that you kind of have to go through when you're using citizen science data.
yeah, cool. Well, so it seems like we don't have any more questions, but I'll wait one more minute just in case.
Make that easier.
Yeah. All right. Well, no other questions are coming in, but I'm sure people could email you or reach out if they have any other additional questions. But thank you so much for sharing today.
Yeah, thanks for inviting me.
All right. Well, have a great weekend, everyone. All right. Bye.
Bye.